A line. A marker. Somebody wrote on the screen by mistake. Okay, welcome to the 7 of you who have weathered the roads. It's not fun out there. The good news is for those who are home in your pajamas, you can listen to this tonight or tomorrow. We have an exam, of course, next Thursday, or this coming Thursday, right, a couple days from now. That exam is going to cover 3 chapters, 17, 18, 19. 17 is the blood, 8 is the heart, which will finish up the last couple of things today, and 19 will cover the majority of today's presentation on arteries and veins. You've seen all this information largely in lab already over the last 2 weeks. Much of this, I hope, is feeling very familiar to you and is somewhat review with some new added details to flavor it a little bit differently. Your vocab on that will be, is it 73 through 92, and I owe you the last 4 or 5 of those vocab slides today. I'll finish up with the heart today and then get through the conversation on the vessels. Just about the exam, make sure that you finish your 3 mastering homework assignments, 1 for each chapter. Those are due anytime before the exam itself. Remember, though, there's also going to be a quiz on mastering that is to help you prepare for this exam, and that'll be due by tomorrow night at midnight. So you've got till tomorrow night at midnight to do your take-home quiz, or I shouldn't say take-home, but the quiz. The take-home exam is already behind us and done. But the quiz, as well as the mastering assignments. And you'll need a Scantron form and your pencil on Thursday. It'll be a full-length exam. Then when we get back next Tuesday, a week from today, we'll have a discussion on the respiratory system. We'll go off and have turkey. We'll come back for 1 week of classes, and we'll have a urinary system lecture on that Tuesday, and on that final Thursday, reproductive systems. And those 3 lectures, respiratory, urinary, and reproductive, will make up the content of the final exam, which I have been told now is on Tuesday of finals week. So that Tuesday, if you're in the morning lecture, it'll be at 10 o'clock in the morning. If you're in the evening lecture, it'll be normal time, 4:30 on that Tuesday. That may be the very first day of exam, so maybe one of the first exams you take if you have other classes.
Do you know what day that is?
Tuesday, is that the 9th? It seems like the 9th. Tuesday the 9th, okay? That sounds right. That'll be Tuesday the 9th at the 4:30 time. And it's just another exam, not a cumulative exam, just another exam over those last 3 systems. Any other logistical things? Remember that your short papers are due before you go away for Thanksgiving. So next Tuesday in lecture will be probably the last opportunity for you to get that to me. You can put it in my box today, all through the week. You can put it in my box next Monday or anytime next Tuesday. When I pull out for turkey weekend, it's late. So make sure I have it before then. Okay. Let's go through vocab. And I think this is where I need to pick up with you. Tempo. If you are keeping tempo, you're keeping the time, if you will. Tens, stretch, tension, stretch, tension. That's how I think about it. Tetan. If you have tetanus, you have a stiffening of the muscles. Tetra, kind of a fun little game, right? 4 squares. And therm, heat, thermometer measuring temperature changes. A little message down there that's not happy. Let's make this happy. Then thorax, chest, thoracic cavity. Thrombo. Remember, platelets are also called thrombocytes because thrombocytes, platelets, are involved with blood clotting. So thrombo refers to blood clot. Tomy, referring to a cut or an incision. So if you see the word appendectomy, you see the tomy at the end telling us it was cut or removed. Tonic, pressure or tension. Remember we had hypo and iso and hypertonic. And that was dealing with water moving in and out of cells based upon solute concentrations. But there is a, quote, pressure, an osmotic pressure that is involved with that tonicity. Tono, again, force or pressure. Topo, place or position. An ectopic pregnancy is one that's out of place. The embryo was trying to set up shop in the fallopian tube. And it's out of place, out of position. Toxins are poison, poison. And tract, draw or drag. So I picture someone's in traction. They are they've had a bone that's in the wrong place, and it's in the healing process, and they'll be in traction. They're actually pulling or dragging those bones back into their proper place. So that's one way to maybe think about that term, tract. A transdermal patch, something that would be a medication infused in a patch. You stick it on your leg or under your arm, and that medication would then go across your skin, transdermal. That's one way of thinking about it. A transatlantic flight going across the Atlantic. 3. Tripsy, crushing. If you have a kidney stone, they might try to put you in a lithotripsor, basically sonicate that. Litho means stone, trips to crush, so to crush the stone and let it pass naturally. Tropic, I wish it was tropic. Tropic is to influence. So some hormones are said to be, as the example says, gonadotropic. That means that they would influence the gonads. So you'll see that word tropic when dealing with hormones and even medications. They affect or influence only certain organs. And then finalizing for today and for this exam, trophy and nourishment. If you have a cast on a arm or leg for a while, the muscles will atrophy. They'll get smaller, and one portion of that is that they're not quite getting the same nourishment. They're kind of wasting away a little bit. Tunica, we've seen that term before, layers, coats, going around the blood vessels. We'll talk about those today. Ultra, beyond or excess. And finally, for this exam, uni, 1. Unicellular, unicycle, whatever, something with uni in it. That does not leave that many slides. It's going to leave about 9 or 10 slides, and those last slides will be part of the final exam. So we're getting awfully close. We're in the U's, and I hope to see great success on the vocab portion of this exam. Now, I owe you just the last little bit of the heart. Now, I actually owe you less than it would suggest from the slides. And this is actually, remember, chapter 18 in the new textbook. I'll get those numbers changed eventually. And as we go through this, let me just flash through some slides. And if something comes up that you need to have clarified, please let me know. So we talked about the overall functions of the heart, not to memorize the numbers, but just to be in awe of this thing in our chest that pumps the blood. We talked about arteries versus veins and the fact that the heart is pumping that blood in 2 different circuits, the pulmonary and the systemic circuits. The pulmonary circuit, just the blood back and forth to the lungs. The systemic circuit going to all of your systems, the rest of the arteries and veins in your body. And you should be able to, with great confidence now, trace the blood through the heart in an image like this. I have an image similar to this at the very end, the very last slide of today's presentation as well, just as a review. Or any image like it. You should be able to take your finger and trace it from right atrium, through the valves, through the chambers, through the vessels, and know the big idea here. Also keeping track of the color flow through the heart. Talked about the overall anatomy of the heart, that it's a little bit off to the left side, that it's in the mediastinum, that there is a layer around it called the pericardium, which is actually a triple layer, the fibrous pericardium, and then the parietal pericardium. Then the serous, the final layer is the visceral pericardium. The heart is made of 3 different layers. We did a quick reminder about cardiac muscle, and we looked at the external anatomy of the heart. And I'm thinking that's about how far we got. Is that accurate? Did I get to the fibrous skeleton of the heart with you guys? Did I get to the fibrous? I'm having a little bit of a senior moment. So we look at the structures of the heart. Most of these are from the lab, right? The things you've already seen in lab. And the fibrous skeleton, what did it do? Gives the heart a structure, right? A foundational structure. And the fibrous skeleton is connective tissue. It's everything that's white in this image. And that's keeping the valves in their proper place and creating a barrier between the atria and the ventricles. And also is an electrical divider, and we'll talk about the cardiac cycle here in a moment, but you want your 2 atria to squeeze, and then your 2 ventricles to squeeze shortly thereafter. You would not want all 4 chambers of your heart squeezing at the same time. That is not a good idea for blood flow. So we want that separation for anatomical reasons as well as the electrical separation. Now, here is where I'm going to do a big flyover. And the flyover, because these are similar to the slides you saw in lab. And it was during the next 12 slides or so, that I would have drawn out the square heart, or where your lab instructor would have given you some information about the heart. You know that it's 4 chambers, the 2 atria, the 2 ventricles. You know that there are valves in place. You know the major vessels dumping back into the right atrium. Right? You've got the inferior vena cava, the superior vena cava, and the coronary sinus. Again, if I say anything that you need to have clarified, let me know. And there are valves. The first valve you would come to would be the right atrioventricular valve, also called the tricuspid valve. And if we go back and look at this image, there's no doubt which valve is the tricuspid. Or really, there's no doubt which one's the bicuspid for sure, right? If I look at this, the bicuspid valve is the one in the upper right. There's no doubt about it. It only has 2 points to it. That is the left atrioventricular valve. The tricuspid valve, you see where it gets its name, 3 points to it. I will be the first to agree that the semilunar valves don't look anything like half a moon, but they do have sort of 3 parts to them, fortunately or unfortunately, when you're trying to learn this. So we got the right AV valve, the tricuspid valve, down into the right ventricle. Here in the ventricle, we're going to see those papillary muscles attached to the side of the ventricular wall, and the chordae tendineae, which are going to help anchor the valve into place. To leave the right ventricle, one is going to go into the pulmonary trunk, and to get into the pulmonary trunk, you must pass through the pulmonary semilunar valve, or just the pulmonary valve, and you'll hear some physicians call it the pulmonic valve. But that valve going into the pulmonary circulation is going to be carrying blue blood. That pulmonary trunk is going to split into the right and left arteries, and it's going to go off to the lungs. Now, let's just take a look at this image of the heart, and I'm just going to point and quiz you on a few things. This is very similar to the heart that you labeled in lab, the colored in, right? So I just put a little dot on it, and you tell me. This is like a little lab practical. I'll put little green dots on it. How's that? Okay? Little green stickers. So, aorta, ascending aorta, or I'd just be okay with aorta, honestly. And I'm going to really get you. Brachiocephalic trunk. Left common carotid. Left subclavian. Pulmonary trunk, which we then split into the pulmonary arteries, left and right. This chamber, right atrium. This vessel, superior vena cava. This vessel, inferior vena cava. This oval indentation, fossa ovalis. This little opening, there's no name for it, but that's the opening for the coronary sinus to bring that blood back from the heart circulation itself. Name this valve. It's the first one, right? Tricuspid, right? This valve, bicuspid. This valve, which semilunar? Going into the pulmonary trunk, so that is the pulmonary valve, or the pulmonary semilunar valve. And the valve hiding in the background is the aortic valve, right? There it is. These muscles on the wall are, let me get them. Stop. That muscle would be a papillary muscle. The point of the heart is called the apex. I know that when I poke into the apex, I have a much thicker layer of muscle. That is the myocardium, and I know if it's really thick that I'm in which chamber? Left ventricle. Good. These red structures are pulmonary veins carrying red blood back to the left atrium, and this guy over here is another one of the pulmonary arteries. Okay, we killed that pretty well. Good. Okay. Not much else I could ask. I wouldn't ask you to label this off a cadaver heart like this, but you should at least recognize the 3 structures coming off the aortic arch. So there's no doubt that this is the aorta, right? Because you see the 3 branches coming off. The one in the front would be the pulmonary trunk. Okay, so we've gone to the lungs, we're coming back, we're in the left atrium, and this story we know, right? Down through the left AV valve, down in the left ventricle. This valve is also called the bicuspid or the mitral. Again, we see the chordae tendineae. We're now in the left ventricle, very thick layer of muscle. Out the left ventricle, we're going to see those papillary muscles and chordae tendineae, and out the aortic semilunar valve. Good with that? Okay. Of course you are. You're the students who are always here. Thank you. Now, let's take a look at this cartoon. This is a new cartoon for you. And if you cut through the heart at this particular angle, now this is a little bit idealized, but really a very easy thing to see. The first thing you recognize, and this is going to give you the 4 valves. Nothing here that you haven't already seen or heard in other places, so you don't have to worry about any extra details in here, but it just goes through the 4 valves, the right AV, the pulmonary semilunar, the left AV, and the aortic valve. It reminds you which chambers or what structures it's between, and really nothing else. And all of them are preventing backflow, right? They're all preventing backflow to the previous chamber. Now this is a flow chart, something you've seen before. Some students will draw sort of a square heart type thing out, some simplistic model of the heart. Others would draw out a flow chart like this. When you get your test on Thursday, I highly recommend it. Have practice kind of drawing out this little flow chart, turn the test over, find some white space, kind of draw out something like that. Then you'll be able to refer back to it throughout the entire test. And you'll find that by knowing the structures of the heart, understanding the flow of blood through the heart, you'll be answering a lot of questions based upon that knowledge. Okay. Now, what this figure shows you, of course, is that blood is coming into the right atrium, as we've done, through the right AV valve. The other name for that, of course, is the tricuspid. Then the right ventricle, the semilunar valve, into the pulmonary trunk, into the pulmonary arteries, off to your tissues for gas exchange, back through a series of, sorry, I just misspoke, to your lungs, right, for gas exchange, and then back through a bunch of pulmonary veins that are red, left atrium, left AV valve, bicuspid or mitral, left ventricle, aortic semilunar valve, the aorta, off to your tissues now, and then back through a series of veins, which come back through your superior and inferior vena cava. Only that's missing over here is, of course, the coronary sinus is another route into the right atrium. So again, having that flow chart in your head, well established, and if you got it, you got it. You don't need to write it down. But if you have any doubt at all, or you're the kind of person like me who would rather just dump it on the test and then be able to kind of quote forget about it and refer back to something that you have, you might enjoy doing that. Plenty of time. There will not be a test 4 again this semester. Okay? This exam will be a far more traditional exam. You won't be crunched for time if you know your stuff. The last test, even those who knew their stuff felt very rushed. Granted, I understand that, right? Overall, though, with the extra points on there, you all did well as a class. But this test will be back to more of a traditional exam. I forget the exact number of questions, but you won't feel quite so pressured at all. Okay. Now, knowing your flow of blood through the heart will be important to understand the cardiac cycle. Now, this will be a major, major component of next semester's course, but we're going to introduce it right now. That is, you have a cardiac cycle. The heart, you know, is beating 75 or so times a minute. It's going to squeeze and relax, squeeze and relax. Each of those cycles is a cardiac cycle. And your chambers are going to be going through alternating times of contraction and of relaxation. During those squeezing times, the blood is going to be pushing open the valves. Okay, I want you to realize that those valves in your heart are not opening and closing like some automatic cuckoo clock, right? Those valves in your heart are opening only when the pressure behind them is sufficient and the blood is being pushed through those valves, more like saloon doors, right, that you're pushing open. Okay, so let's take a look at this cardiac cycle. Now, there's 2 words on here I want to introduce. The 2 words I want to introduce are systole. You know from your vocab, systole or systolic means contraction. This is the time during which the heart is squeezing. And then there's diastole, the time during which the heart is relaxing, or in between the times of a contraction. Now, when the chamber is in systole, it is squeezing, pushing blood into the next place. When the chamber is in relaxation or diastole, it is during that time that it is refilling with blood. So we have this, we're imagining this ever-moving blood being pushed through these chambers. Now, on this figure, first thing we're going to do is cross off everything over here on the right-hand side. So we're not going to worry about any of that. What we're going to focus on is just the left-hand side, and I know this is small, but let me walk you through this. So first thing we notice is that over here on the left it says structure. Atria, ventricles, AV valves, plural, left and right, semilunar valves, plural, both the pulmonary and the aortic valves. First phase, atrial systole. That means your atria are squeezing. Okay, so if the atria are squeezing, does it make sense that the atria are contracting? And when the atria are contracting, the ventricles aren't, so the ventricles are relaxing. When the atria squeeze, what's being pushed open? The AV valves. Can you picture it? And then, when those are squeezing, the semilunar valves would be closed. Okay, they're taking turns. Now, what's going to happen? Now, this is stretched out, but this is when the ventricles are squeezing. Ventricular systole. Okay, it makes sense, I think. When the ventricles are squeezing, right, the ventricles are contracting, the atria are relaxing. When the ventricle is squeezing, it's going to be pushing open the pulmonary and the aortic semilunar valves. This is critical. When the ventricles are squeezing, the AV valves better be closed. Because if the AV valves weren't closed and the ventricles were squeezing, where would blood be going? Backwards, right? Back up into the atria. And when blood goes backwards, our heart's not working efficiently, and we call it a murmur. So when blood is going backwards through one of the valves improperly, that's a murmur. Now, a little bit of a murmur, okay, we kind of follow it along, and we say, oh, you've had a murmur since you were born, and we kind of follow it. But if that backflow becomes sufficient or significant, then it can really start decreasing the overall efficiency of the heart, and you might have some valvular issues you got to address surgically. So we're not going to go over to the other side, right? We're not going to worry about this whole thing over here on the right-hand side. Just focus on what I just said. And keep in mind that as the chamber is squeezing, it's going to squeeze, increase the pressure, and push open that valve. And now the blood will squirt into that next place, right? So from the atria down to the ventricles, from the ventricles, push open the semilunars, and out either the pulmonary trunk or out the aorta. So this constant squeezing, relaxation, systole, diastole, systole, diastole. What do we think? Make sense? Can you picture it? Knowing the structures and the valves, just picture this basic idea of the flow of blood through the heart. Now, the heart is autorhythmic. And what that means is that you do not have to have your brain attached to your heart. The heart will beat on its own, even with no brain attached. There are cells within the heart itself, specifically located in the right atrium. Okay? There are cells in your right atrium called your pacemaker. You've heard of this term, right? Pacemakers. People have an artificial pacemaker put in because their heart no longer does this properly. So within your right atrium, within your right atrium, there are cells called pacemaker cells, and they are actually sending the signal to your heart to contract. If that part of the heart gets damaged, we can put in an external, fake pacemaker, if you will, and that will be the backup system. That pacemaker, those cells are going to send electrical signals and tell the heart to beat. Again, a person can be completely brain dead. You can disconnect the heart from the brain, and the heart will keep on beating in a transplant kind of situation. And that's all I'm really going to say about that. We'll deal with that whole pacemaker deal next semester. Now, the heart is connected to your brain. I'm not suggesting that it's not. What I am saying is that you don't need the brain for the heart to beat. But you do need input from the brain to modulate your heart rate. So we've appreciated already that when the bear chases you, your heart rate goes up. So under sympathetic signals, your heart rate goes up. Under parasympathetic signals, your heart rate goes down. You can't go down too low, but it will calm down a little bit. So we see that the heart is affected by the nervous system, but it is not initiated by the nervous system. Okay, so the initiation of the heartbeat is by the heart itself from the right atrium, called the pacemaker, and the heart is influenced by nerves going to the heart. Now, the nerves going to the heart collectively would be called the coronary plexus. Remember, a plexus is a group of nerves that travel together. We've seen the brachial plexus and the sacral plexus. We know this term. So there's a group of nerves that go to the heart. Collectively, they are the coronary plexus. Same idea, the nerves going to the kidney would be called the renal plexus. So we'll see this again. We already know how sympathetic and parasympathetic affect the heart. Now, lastly, we're going to, just like we did in lab, we're going to skip over specific names of the arteries that go to the heart. We're just going to call them the coronary arteries. They have names. We're not going to learn them this semester. We crossed them off in the lab as well. So you know that as soon as your blood comes up out of the aorta, you'll see on the models that there is almost an immediate branching off from the aorta going to feed the heart itself. Those are the coronary arteries. There's a major left and a major right coronary artery. Now, that coronary artery is going to split into smaller arteries, which we're not going to name. Okay, but there's a marginal and a posterior interventricular artery. You can even picture where that darn thing goes, right? On the backside between the ventricles, right? Posterior interventricular. But you don't have to learn those. Then on the left side, likewise, there is branching, and we're not going to worry about the individual names here either. Here is basically a diagram of those coronary arteries. And you can appreciate, here's the aortic semilunar valve. And as soon as blood comes up through that aortic valve, there's your branching, right? So your coronary arteries, left and right, are coming right off. This is the freshest, most oxygenated blood available to the body. That blood is going to go around and feed, bring oxygen and nutrients to the heart muscle. If you had a blockage down here, see where I'm drawing this? If you had a little blockage way down there, and so only this little part of the heart did not get oxygen, and you had a heart attack, right? There's no such thing as a small heart attack, but it would not be a major heart attack. Only that small part of the heart would have lacked oxygen. That small part of the heart would not recover. Cardiac muscle does not redivide, does not undergo mitosis, does not repair itself very well at all. That part of the heart would die, but that person would very likely survive that heart attack quite well. It's a very insignificant percentage of the heart that's been affected. However, if that blockage was up here, and now you have blocked all of the right ventricle from receiving blood, that would be, quote, a massive coronary, massive heart attack, and likely the person would not survive. So it's all about how much of the heart is affected by the heart attack and the timing of it as well, right? If we get quick help or if that heart muscle is completely destroyed in the process. So we see the coronary arteries, just name them the coronary arteries, left and right. And then the next image shows you the coronary or the cardiac veins. So once that blood has fed the myocardium, now all that blood comes back through a series of cardiac veins, and those cardiac veins all come back and drain into the coronary sinus, and we can see that the coronary sinus has a little opening directly back into the right atrium. Are we good with that? Now, on this test, you can certainly expect that someone's going to need a blood transfusion as a case study type idea. So you know someone's going to need a blood transfusion. You better know from whom and to whom they can give blood. In fact, after that blood transfusion, they're going to be so thankful they're going to want to go down to the Red Cross and donate blood. And so you need to know to whom they can give blood. So expect that kind of question in a case study. Also, someone's probably going to have a heart attack. So this is what I want you to know about a heart attack. Okay. A myocardial infarction. It is a sudden and complete blockage of one of the coronary arteries. Okay. So this is all part of the coronary heart disease story. And what leads to this blockage is typically some sort of atherosclerosis. Atherofat, sclerohard, osis condition of. So a condition of the hardening of fatty deposits around the heart, around the vessels. Now that can lead to one of two things. Now there's a lot more than this. But an MI, a myocardial infarction, a complete blockage, or it could, as it's beginning to clog up the artery, lead to what's called angina or angina. You'll hear it pronounced both ways. Now we don't hear about angina very much anymore, because now if a person has angina, right, so you're going to hear more about angina in the news because people are going out and shoveling their snow, and they're going to be exerting themselves beyond their normal level. So Grandpa Joe's going to go out and shovel his snow, and it's going to cause an increase in the heart production, and maybe his heart can't handle that, and he's already got some blockages to some of these vessels that are undiagnosed. And he's going to have some localized pain as if he's having a heart attack. But it can be treated very easily with nitroglycerin. Now, nitroglycerin is a little pill that people with angina carry with them, and they pop it under the tongue. It very quickly is dissolved and absorbed into the body. And nitro is a vasodilator. So it opens up the vessels, including those coronary arteries, and allows the blood to flow now more normally around that slight blockage. And they go on, and everything's fine. Now, a person who has angina nowadays would likely go in and have a stent put in, some chicken wire put into the vessel, or angioplasty, or something, or a bypass surgery, something that they would never have to deal with that slight blockage. But that was very common 20, 30 years ago, or people who don't go for good care. They may have these intermittent attacks, mini blockages. Now, versus a heart attack, a complete and sudden blockage of one of the coronary arteries, the myocardium is completely deprived of oxygen. That region of the heart muscle dies very quickly. This is not a cell or a tissue that likes being without oxygen, and they don't regenerate. So the scar tissue that is left behind does not contract, and so the heart is no longer as efficient as it was. So just be able to differentiate, if you will, angina from a heart attack. And again, you might have a case study that mentions one of those two conditions, and you would be able to appreciate the difference between them. The last slide is something, again, you should be able to appreciate and trace along in your mind or with your finger. The 11 steps on the left are the same 11 steps that you should now be very comfortable with as you bring blood through the heart. Everything on the right side is blue, everything on the left side is red. You know your vessels, you know your valves, you know your chambers. What can I entertain? Any questions on the heart at all? Or blood, going back to blood. Yes?
I think I have with the engines of giving and receiving blood.
Okay.
I can figure all of that out. I just can't remember.
So the question is back to blood typing?
That you can donate to.
Yeah. Okay. Let's just do this really quick. I'll do a blood typing really, really quick, okay, on the white screen here. And so the question was just kind of go through the blood typing story really quick, right? So let's go with A negative blood. And this is just the way I always do it. So you've got an A negative red blood cell. This person has inherited what from their parents? What antigens are on the surface? Just A. They did not inherit Rh. They did not inherit B. Because they don't have B or Rh, their body would consider both of those foreign and would now make antibodies against B and against Rh. Right? They're going to make antibodies against B and against Rh. Because they're making antibodies, they could not receive blood with has B in it or Rh. So that means inward arrow X, not able to receive anything with B in it. That'd be the B positive and the B negative. The AB positive or negative, again, they all have what in them? B. And nothing with Rh, so I also couldn't receive A positive or O positive. All 6 of those have either B in their name or Rh in their name. If they were received by this person with A negative, agglutination would occur, clumping. What's left? There are only 8 blood types. So this person could, with no problem, receive A neg and O neg. That's all that's left. Now, A negative, the same blood type, no surprise there. O negative, the universal donor, so you're not surprised to see O negative there either. Now, when you go to give the blood away, right, outward arrow. When I give my blood away, what am I giving away? A, right? The person's going to be receiving my blood that has A on it. So that means their immune system must be ready to receive A without issue, which means that they themselves already have A. So which blood types already have A? A positive and negative, AB positive and negative. They already have A. Their immune system says, no big deal. I'm used to A. Bring it on. Okay. So my recommendation, I just, the way I do blood typing is I like to think through this kind of series of questions, this logical progression through it. What antigens are there? What antibodies would be produced? If I know what antibodies are being produced, I know what I cannot receive. Then I can blindly fill in what I can receive, the rest of them. And then when I give my blood away, I'm giving away the antigens on the surface of my blood, so I can only give it to people whose body already has that antigen, because their immune system says I'm cool with that. To help clarify, quick little reminder about blood typing. And there will definitely be, in addition to the case study on the exam, there'll be a couple of other questions that say, if you have A positive blood, to whom and from whom can you receive blood? So it is an important concept that you need to understand for this test to get all those points. Anything else to clarify? Blood or heart? Okay, the last little conversation we have is over the last chapter of this test. It is going to be over the blood vessels. This is chapter 19 from your Martini book. And as has already been sort of the theme, we know a lot of this. So some of this will be review, I hope very straightforward, and I will kind of wake you up when we get to something brand new. Let's think about this. Blood vessels, we know what they're doing. They're sending blood around the body. In that blood, there is dissolved oxygen. There are nutrients. There are hormones. There are waste products. All sorts of things are being transported through the body, through that blood. It's being pushed around by the heart. This is a closed loop system, and this blood is constantly moving and supplying and picking up things from our tissues. However, this is not a rigid system, and you know this from our conversation of the sympathetic and parasympathetic nervous systems. You know, for example, that the body only has about 5 liters of blood, and that when the bear walks in, the body says, you know what, I need to send more blood over to the skeletal muscle, and I'm not going to send as much to the stomach. And when you're in the rest and digest, the body says, okay, right now I don't need to worry about running away. I need to worry about sending more blood to my digestive system to digest my meal. So the body is making choices. So while, yes, the arteries and veins are in place, they are constantly changing to meet the needs of the body. They're pulsating, and they are carrying more or less blood based upon the needs. Now, you know this as well. You know that the naming of the vessels takes a little bit of time to look at them, but the names here are not new. In fact, the only new names that are in this list are the word jugular, which are the veins that bring blood down the neck from the brain, and carotid, which are the arteries that carry the blood up the neck to the brain. Other than that, all the other arteries and vein names are body regions or terms that you've seen. So if you would just spend a few minutes, the arteries and veins that you need to know for this exam are no different than the list you had to know for the lab. So on the back of your pre-lab number 9 or 10, whichever one it was, there was a list of arteries and veins, and that's the same list that you need to be able to correctly identify for this test. There will be a couple of questions. Name that artery, name that vein. They're not hard. I assure you they're not hard in the test, but don't forget to at least look at that. Those arteries and veins are traveling typically side by side. For example, the femoral artery travels right next to the femoral vein, which, oh, by the way, travels right next to the femoral nerve. They kind of travel as a triad, but they're running along the femur, right? So the naming of them is not too difficult. We know that these vessels are carrying blood through two circuits, mentioned them already today. The systemic circuit, carrying blood to all of your systems, and the pulmonary circuit, dealing with the blood to and from your lungs. They're working side by side, but the heart really is sort of a double pump, sitting side by side, the right side and the left side. And cardiologists will refer to the right heart and the left heart, because although they are side by side, they are really responsible for different things, and things can go wrong in one side or the other. Which side of the heart pumps more blood, right or left? Which side of the heart pumps more volume of blood, right or left? It better be the same. Now, I know that it seems like a trick question, right? Because we know the left side is definitely more muscular. And the left side definitely has to pump with more pressure, more force. But if the left and the right sides were pumping different volumes, we would quickly have a backflow or an overflow somewhere, right? So both sides are pumping the same volume. Yes, they're pumping under different force. And we'll deal with that again more next semester. I know I keep saying you can't wait to get to next semester, right? Because we're going to finish the story. We're going to talk more about it. But we'll talk more about how that whole thing happens. And you know that the heart is pumping blood out from it through arteries. The big two arteries are the aorta and the pulmonary trunk. I had a student today in lab. I was very pleased. But after 2 weeks of cardiovascular, talking about the heart 2 weeks ago and last week talking about arteries and veins, today after the artery and vein quiz, she said, oh, arteries are usually red. Yes. So it's good that these light bulbs go off, right? It just took a little longer than I expected. So maybe some of you are still at that point where you're like, oh, okay, there are connections here you need to make. Once those connections are made, this will make a whole lot more sense, right? But it's going to, maybe for some of you, you're still dealing with, blood typing a little bit or arteries and veins and trying to get the whole idea here. Now, the arteries are going to move away from the heart. That's always, always true. No exception. Always going away from the heart. Those arteries are going to split. And many of the arteries that split off from the aorta are the arteries you're naming, right? The common iliac, the internal external iliac, the carotid, the brachiocephalic. All of those are branches off from the aorta. They're getting smaller and smaller. Eventually, that blood is going to enter into capillaries. Those are very, very fine, small, microscopic vessels. That's where all the gas and nutrient exchange happens, the fluid exchange, everything happens there. And then that blood's going to come back through a series of veins, and those veins are going to get bigger and bigger and bigger as they come back toward the heart. That we should get. Now, here's a term that I want you to at least appreciate, and that is the term anastomosis. An anastomosis is used in a couple different ways in medicine. One is where two vessels come together, or two vessels merge. Another way of thinking about it, though, is alternate route. Okay, anastomosis, aka sort of alternate route. And here's what I want you to think about it. It tells you in the bottom that veins tend to have more anastomoses than arteries. Have you ever heard of someone having veins stripped out of their leg? Maybe they have varicose veins, or they've got painful veins, spider veins, and those veins can become painful and certainly unsightly, and the person will have them stripped, taken from their leg. What happens? The blood still gets back to the heart via a different route, right? There's another way for the blood to get back to the heart. Now, if you took an artery out of your body, you'd be in trouble, because the artery, there aren't many ways that blood gets, to example, to different parts of your body. There's an artery that carries it there. If you took the artery out, there wouldn't be another route for blood to get to that tissue. So there are more venous anastomoses, more alternative routes for blood to come back to the heart through your veins than there are for your arteries. And that's really what I'm trying to tell you here in this slide. Now, here's something that you have already picked up from in lab, I hope. That is that vessels have 3 layers, or at least most of them do. Arteries and veins, 3 layers or tunics. tunica intima or interna is the inner layer. It is structurally only made up of a simple squamous epithelium. You remember what that looks like, right? One layer thick, very flat cells. This is not a very thick layer, very delicate. The name for this layer is the endothelium. It's a special name. Whenever you hear about the endothelium, it's referring to the lining of the inside of a blood vessel. The middle layer of the vessel is the tunica media. Middle, media, muscle, all the M's. This is smooth muscle. It is involuntary muscle, but this is where all the control comes from that I discussed in the sympathetic and parasympathetic nervous systems. So when the bear walks in the room and more blood has to go to your muscles, the blood vessels open up to your muscles. They vasodilate. Vas means vessel. Your vessels dilate, bringing more blood to that area. At the same time, though, your blood vessels going to your stomach are going to vasoconstrict. Again, this idea of the body meeting your immediate needs through the opening and closing, or at least the constriction and opening of these different vessels. All that control of opening and closing vessels happens because of the muscle in the tunica media, this involuntary muscle that responds to hormones and other signals. And then finally, the outer layer is made up of fibroblasts. Those fibroblasts are making collagen. It's a connective tissue layer. They're making collagen that's going to give vessels their strength, and it's making elastin, giving them their stretch, their elasticity. Now that makes sense to you, it should. When you take someone's pulse and you put your fingers on your neck and take your carotid pulse, or take your fingers and put them on your wrist for the radial pulse, what are you feeling? Pulsing of the blood going through your artery. You got to push a little bit hard, right? It's not right at the surface. You got to push down just a little bit. Remember, arteries are a little bit deeper than veins. So you got to push down just a little bit, push in your neck a little bit. You'll feel that pulsation of the arteries. So it makes sense that there's going to be some elastic properties to your vessels. This outer layer is also going to anchor this vessel, these arteries and veins, to your body walls and to your organs. They're not just these tubes running through your body. And finally, they're living, right? When you think about a blood vessel, I don't think of a living thing. I think, oh, it's just a straw, a tube, a hose going through my body. But remember, on the inside, it's epithelial cells. In the middle, it's smooth muscle cells. On the outside, it's collagen. It's fibroblasts. Those cells themselves need to be nourished. And the blood going through the vessel provides some nourishment, but there also needs to be a separate supply of blood to the cells that make up the vessel. So in the outer layer, in the tunica externa, there's a very fine network of small arteries and veins that are going to carry blood to and from the artery itself, or the vein itself. And this is referred to as the vasa vasorum, vessel of the vessel. Vasa vasorum, vessel of the vessels. Now, before I move on to this next slide, I want to tell you that on this exam, you're going to see a Venn diagram. Did you like my Venn diagram on, remember we did one on the sympathetic and the parasympathetic nervous systems? That's what we did last test. There were like 10 questions you had to know, or maybe 6. This test will have a Venn diagram. It'll look like this. This will be for arteries. This will be for veins. This will be for capillaries. Okay? Now, artery for A, C for capillary. The vein will be the letter B. Okay? And I'll make it very clear what's going on. And then I'll have some factoids down here, some facts. 1, 2, 3, 4, 5, 6. So as I'm going through this in the next few minutes, what you need to be really capturing is the comparison and contrasting of the arteries and the veins and the capillaries. What fact is true of just arteries? What fact is true just of veins? What might I say that it pertains to both or to all of these kinds of vessels? And there will be this kind of Venn diagram situation on your test. So let me help you with this. Let me give you some of these facts and show you exactly where in your notes right now you would go to answer this. And then I'll quiz you on this when we're done. So, arteries. In arteries, the tunica media is the thickest layer. Why would that be? Tunica media is your muscle. Why would an artery have more muscle than other vessels? Has to withstand the pressure, doesn't it? You don't want to go to Lowe's and buy a cheap hose, right? When you go to Lowe's, you buy the cheap hose, the medium hose, and the expensive hose. And the expensive hose is more expensive because it's a thicker wall. And you want that thick-walled hose if you want to hang on to it for a few years. If you want a garden hose that's disposable, buy the cheap one. So you want a thick layer of muscle, a really thick layer. It's going to withstand the pressure better in the hose. So that kind of makes sense, right? Veins don't have a lot of pressure. And I'll keep saying that over and over and over. Veins don't have a lot of pressure. Therefore, or they don't have a need for a lot of muscle. They instead, their outer layer is the thickest layer, the tunica externa. Okay? So that's one difference between arteries and veins. Number two, if you had an artery and a vein traveling side by side, imagine a red pencil traveling next to a blue pencil, right? And from the outside, they would have about the same overall diameter. But if you cut into them, you would note that the artery is very muscular and has a very thin lumen, a very small lumen, a very small opening down the middle of the artery, whereas the vein has a much bigger lumen. Now again, I think that kind of makes sense. Arteries must maintain the pressure to get the blood all the way from the heart to the brain. And to help maintain that pressure, you want a thick muscular wall and you want a very small lumen. You want to keep that pressure high going through that tube. If you squeeze on a hose, right, the pressure goes up. So you want to keep that pressure high. You want a very small lumen. Whereas in a vein, you're going to see the lumen is much bigger. The lumen is not under a lot of pressure and more blood will fit in a vein than in an artery. Number three, arteries have more elasticity. And again, that makes sense. Arteries are under a lot of pressure. As blood leaves your heart and goes into your aorta stretches and recoils. And we feel that same thing when you take your pulse. Your carotid arteries are recoiling and, stretching and recoiling. So we know there's some elastin that provide that characteristic of your arteries. And that would not be a characteristic of your veins. So your veins would not be elastic. Okay? In fact, they tend to spread and keep spreading. They don't fight back. An artery, if you push on it, will kind of push back at you. It's very muscular, very elastic. If you push on a vein, it just kind of collapses. There's not much structure to it. There's not much muscle to it. It doesn't spring back at you because there's not much elastin in it. In fact, that's what causes varicose veins. They get full of blood and they just spread, right? They just kind of spread. They just kind of spread out. They don't have a muscular wall. Another important distinction is that capillaries don't have all three layers. Capillaries only are composed of the tunica intima, that inside layer of the simple squamous called the endothelium. That's really all there is to a capillary. It's very, very delicate. But we know that what happens in that capillary is exchange. Simple squamous, also found in your alveoli, right? So that lesson from histology starts to make sense to us. In the alveoli, simple squamous, gas exchange. In the capillary, simple squamous, gas exchange. Form and function go together. Now here's a beautiful table. I know it's small, and there's nothing on here that I haven't already described to you. But if you want all these factoids in one place, this is a great table to pull up maybe on your tablet, computer, or whatever over the next couple of days and just stare at it for a minute. And the facts that I will take about arteries versus veins versus capillaries will come largely from this table. So if you look at this, the lumen diameter, right, more narrow in arteries and veins. Wall thickness, thicker in arteries. Cross-sectional shape keeps its shape in arteries. Veins tend to collapse. Thickest layer, tunica media in the artery. Elastic qualities, strength and elastic qualities, more in the artery. Pressure, greater in the artery. Flow, always away from the heart in the artery. And then lastly, what color is the blood? Oxygen levels, remember there's a difference here. In your systemic arteries going off to your tissues, those are going to be bright red, high in oxygen, whereas your pulmonary arteries are going to be lower in oxygen. So this is a nice table just to keep that compare and contrast story together. So going back, if I said to you, oh, here's a nice picture of everything I've said, and then I'll show you the Venn diagram. So here we have an artery and a vein side by side in a picture. If they were closed and sitting side by side, it would be like a red pencil next to a blue pencil. You wouldn't see a difference in the overall diameter. But cutting into it and looking at the layers, you'll appreciate a difference. So again, they all have a tunica intima, and that is just the simple squamous epithelium on the inside. They both have a tunica media, but the muscle layer is very substantial on the artery and very wimpy over here on the right on the vein. The tunica externa, also present in both, not very impressive on the artery, thicker on the vein, like I mentioned. And when you look at this image, you will see microscopic arteries and veins in that external layer. That is the vasa vasorum, the little vessels that feed the cells of the bigger vessels. This image also reminds us that the capillary is not composed of all three layers, but just the endothelium, just the tunica intima. And lastly, something I haven't talked about today, valves are found in your veins. So there are valves in your veins of the legs and arm. And we'll talk about the significance of those valves in a few minutes. So that's a nice little pictorial way. Oh, last thing, I've already said it, the lumen, right? Quite narrow in an artery, more expansive in a vein. Okay, so those facts are all there in a picture. So if I were to create the Venn diagram scenario, and I had the, here we go, artery, veins, we'll call that B, and capillaries, C. And I said something like, has the thickest layer of muscle. On the test, it would be like this, A, B, C. This would be AC, this would be BC, this would be AB. If it applied to all of them, A, B, and C. Okay, something like that. So if I said has a thick layer of muscle, the tunica media is the thickest, that applies only to arteries, so you would plug in letter A. If I said to you, has all three layers, three tunics, that is only true of arteries and veins, not of capillaries. So that would be, you would plug in A and B, right? Bubble in the union of those two. If I said to you, is an important part of your cardiovascular system, all of them, right? Or contains a tunica intima, all of them, then you would plug in A, B, and C. All of them are true. If I said to you, always carries red blood, is that true of any of those? So there would be a little mention in the instructions that letter D would be the answer if it doesn't apply to any of them. Okay. So same idea, Venn diagram, facts about arteries, veins, and capillaries, and that'll be definitely something you should expect on the exam. So be keeping those little facts in mind as we go through this. Always go away from the heart. Arteries, right? Always carry red blood. Not true of anything, right? So we would have the D. So you know the arteries, just a little bit more about arteries. So the arteries are carrying oxygenated blood, except, right? We keep saying the except, in the pulmonary circuit where the arteries are blue. And it turns out there are three flavors of arteries. This has to do with their structure. There are the elastic arteries. Elastic arteries, as the name suggests, have a lot of stretch. These are your great arteries, that is your pulmonary trunk and your aorta. So the big arteries coming directly off the heart have the most elastin because they have to stretch the most because they're under the greatest pressure. Then your medium-sized arteries are simply called your muscular arteries. These are the ones that we're naming, right? This is your brachial artery, your common iliac artery, your renal artery. And eventually those named arteries, those muscular arteries, are going to branch down into very small microscopic arterioles. There are no arterioles that you're naming, right, in this system. They're all just microscopic. Please do not memorize the numbers. Don't be concerned with the numbers. Just know the order, right? Large elastic arteries to medium-sized muscular arteries to the microscopic arterioles. As we move through this progression, the diameter gets smaller, but the actual amount of muscle increases. Arterioles are incredibly muscular. Again, that's where all that regulation comes from, controlling blood flow to different portions of the body. Histologically, I'm not going to ask you to label this, but the middle picture is a medium-sized muscular artery. So the inside layer would be what? What layer? Tunica intima, made up only of simple squamous epithelium called the endothelium. The pink layer, pretty thick, tunica media, made up of smooth muscle. The outer layer, not quite as thick, but around there, tunica externa, fibroblasts, making up collagen and elastin. Okay. So that's all I want you to know about arteries. 3 different flavors, thick muscles, all that good stuff. Now let's move down to capillaries. You know that the capillaries are the linkers between the small arteries, the arteries, the venules. Now I wish it was spelled this way. Why do we have arterioles and why don't we have venules? But we don't. We have venules, right? Those are the small veins, and I'll mention that again in a minute. So venules is wrong, right? Should be that way though, right? Don't you agree? Now, I don't want you to memorize this number, but I want you to appreciate that capillaries are 8 to 10 microns thick. Now, that's significant because how big is a red blood cell? Do you remember this number? This is the number you need to know anyway. Yeah, red blood cell is 7.5 micrometers across. So I just want you to appreciate that the trip of a red blood cell through a capillary is rather claustrophobic. It's a very tight squeeze, okay, through these capillaries. And we'll talk about that again in a moment. You know that the capillaries are only made of one thin layer called the tunica intima, and that this layer is important for the gas and nutrient exchange that occurs. Because this is where all the real action happens within the cardiovascular system, the capillaries are considered the functional unit of the cardiovascular system. We've seen that functional unit idea before. In skeletal muscle, do you recall the skeletal muscle functional unit was the sarcomere. Remember that little sliding filament, actins and myosin sliding over each other. In the nephron, you're going to learn that the functional unit are these series of tubes called nephrons. So that idea, where all the microscopic action happens, is called the functional unit. And you've seen in a lot of cartoons that capillaries don't exist as a single little vessel, but in a cluster called a capillary bed. It's typically between 10 and 100 of these little vessels clustered together. Now this is brand new for you. The capillary bed is fed by a little linker called the meta-arteriole, and I'll show you a picture of the meta-arteriole in a moment. And what we're going to see is that on the proximal end of this meta-arteriole, there are going to be some smooth muscles that act as little sphincters, little circular muscles that are going to wrap around. And what they're going to do is make sure that not too much pressure goes into the capillary. Remember, the blood's leaving the heart under a lot of pressure, right? And by the time it gets down to delivering the oxygen to the capillaries, those capillaries are rather delicate. So we can't have too much blood pressure going into a capillary bed, or else you'll basically destroy the capillary bed. So we have to be able to regulate very tightly the pressure of the blood going through the capillary bed. And we'll see in a picture in a moment these little muscles that are going to help regulate that. About every 5 or 10 times a minute, your little sphincters, these little muscle groups, are going to open and close. And this is going to allow blood to enter into your capillaries. In other words, all of your capillaries are not open at the same time. If all of your capillaries were wide open, there would be such an area available to your blood that your blood pressure would drop. We'll talk more about that again next semester. But you need to make sure that only some of your capillaries are open at any one time. And the body does this through this thing called vasomotion, this on and off switch that occurs. Let's take a look at this cartoon of a simplified capillary bed. So what I see, blood coming in the arrow, in an arteriole. It shows on the outside of this arteriole smooth muscle cells. Remember that little fusiform shaped cell? So the muscle layer is shown here. The external layer has been stripped away for the purpose of this cartoon. The blood's going to travel in this little arteriole and take a little turn into a capillary bed. Now this linker right here that connects the arteriole to the capillary bed, that's the meta-arteriole. This little connector between the arteriole and the capillary. And then notice that wrapped around in the meta-arteriole, we see these little muscle cells. And they're acting as little sphincters, little squeezing muscles. And they're going to be opening and closing and regulating how much blood can come into the capillary bed. Again, if there's too much pressure coming in, it would destroy and damage these capillaries. The blood's going to come through. We see the red to blue transition. Oxygen's being dropped off. CO2 is being picked up. Nutrients are being dropped off. Waste products are being picked up. Water is exchanging. The lymph, the interstitial fluid of the lymphatic system, is interchanging with the plasma, the watery portion of your blood. All that's happening here in these capillaries. The blood then leaves out, having dropped off its oxygen and the waste products, into a series of small veins, and we see now it's called the venule, right? The tiny microscopic veins that drain this blood back. Now, this is also brand new for you. It turns out there are 3 types of arteries based upon size, and there are 3 types of capillaries. You've already seen a couple of them in passing, or you will see them tonight in lab. The most normal, numerous type of capillary that you dream about when you're going to sleep are continuous capillaries. These are your normal capillaries. This is what I just described to you, where gases are exchanged. Then there are fenestrated capillaries. Now, fenestra is Latin for window, openings, holes, okay? And this is the kind of capillaries one sees in the glomerulus. Now, if you've had the lab already, you know that the kidney, right, has glomerulus, the filtering capillaries, and those capillaries have little holes in them, and those little holes are called fenestrations. Now, the other places that you'll see those fenestrated capillaries are in the endocrine glands and also in the small intestine. Just make sense of this. Why would you need to have fenestrated capillaries with little holes in them, in your or around your endocrine glands? Yeah, those hormones, right? Endocrine glands are producing hormones. Those hormones, some of them are kind of large, and they have to get into your bloodstream, right? They are released directly into your bloodstream. So there better be a gate, an opening, a hole for these hormones to enter into your bloodstream. So around your endocrine glands, you'll find fenestrated capillaries with holes in them. And in the small intestine, some of your nutrients, the way I think about it, are kind of chunky, right? You've got to be able to take up some larger molecules into your bloodstream through your intestine. So your small intestine would also have a fair number of these wholly fenestrated capillaries. Does that make sense? And they're in the kidney because of the filtration that's going on in the kidney. Third type of capillary, which we've not discussed at all, are called discontinuous or sinusoid capillaries. These have huge gaping holes in them. It looks like a bomb went off in them. And we're going to find these in the bone marrow, liver, spleen, and also some around some endocrine glands. So why the bone marrow? Why would you need big gaping holes in the capillaries of the bone marrow?
I already watched this earlier.
Why?
It's the blood production and platelets.
Right, in the bone marrow, we're making red blood cells. those red blood cells better have a way of getting into the blood system, right? So we need to have a gate big enough for those blood cells to come in. And how about the liver and the spleen? The liver and the spleen, remember back to our conversation about red blood cells. The liver and the spleen were important for recycling, breaking down damaged and decaying, dying red blood cells. So again, those red blood cells need a way of exiting the bloodstream into the spleen, into the liver, to find their recycling center. And again, some hormones are big enough that the endocrine glands have larger sinusoidal type capillaries. Let's take a look at this picture-wise. This makes it a lot more simple. So on the left, there's your normal continuous capillary, the one that you know and love. Notice there are no gaps or spaces between the cells. I also want you to get the idea of the scale of size. I told you that a capillary is about 8 to 10 microns across, and that red blood cell is about 7.5 microns. So again, you're never going to see, that red blood cell is completely contained within the capillary. Here in the middle, fenestrated, little openings, little holes, only big enough for very small things like, glucose or nutrients, water, maybe some hormones, maybe some nutrients being absorbed in the gut through those little holes. And then here's your big discontinuous sinusoidal. So now you see gaping holes big enough for red blood cells to enter and exit. So that's in your bone marrow, liver, and spleen. Does that make sense as to why you would find those different types of capillaries? They're still doing the same thing, but they are allowing bigger or smaller things in and out, aren't they? Okay, so that's our story about capillaries. Now, let's move to veins. Well, we know that the veins are receiving the blood that just traveled through the capillary. First, these little venules, then bigger veins. And as we've already discussed, the wall of veins are very thin, and the lumen of them is larger. They typically carry deoxygenated blood, blue blood, except for the pulmonary veins, which, as we know, carry the red blood. And the pressure is very low. By the time the blood goes through the capillaries and comes back to the veins, the pressure is very, very low. And we'll talk about how is it that the blood gets back to the heart. Finally, if I were to ask you right now, while you're sitting at rest, what percentage of your blood is in your arteries and what percentage of your blood is in your veins, I would probably guess, 50-50. But in fact, remember I talked about the lumen of the arteries is more narrow, the lumen of the veins is larger. So at rest, you have about 60% of your blood sitting in your veins and only about 40% sitting in your arteries. That kind of makes sense too when you consider if you're sitting there right now and you're kind of resting, but the bear walks in the room and you need more blood now pumping quickly through your body, you better have a reservoir, a place for that blood to come from in that time of greater need. So more of your blood is sitting in your veins at rest than in your arteries. Now again, the venules are your tiny, tiny microscopic veins. Don't worry about the numbers again. And that blood is going to go into veins. Now the pressure in veins is very low. In fact, gravity would win as far as pushing blood or allowing blood to pool in your extremities if it weren't for a couple of things. One, your heart is a pump. Now, pumps not only push things out, but they also suck things back. And so the heart is sucking. It's pulling blood back into the right atrium through these veins, right? So that's something that's going on. Number two, there are valves in your veins of your arm and legs. And those valves, as blood starts coming back, the blood's not allowed to go back. It can't go back toward gravity. Now, this is a bigger issue in your legs than it is in your arms. And then finally, your veins are, with those valves, every time you walk, for example, every time you move your muscles, every time you breathe, your diaphragm and your arm muscles and your leg muscles are squeezing on veins. Most of your blood is coming back through veins that are traveling through the center of your muscle groups. And as your gastrocnemius or as your soleus or as all of your leg muscles squeeze, blood is being pushed up and then it's kept at that level because of the valves that don't let it go back. So you've got the sucking of the heart, you've got the pushing of the blood by the muscles, and you've got the valves making sure it doesn't go backwards. And that's really the way by which your blood comes back to your heart. It's not being pushed, right, by pressure. It's pretty much zero pressure. So if you want to keep a healthy heart, walk, move, do something, right, that keeps that heart having to work less hard. But what happens to a muscle that has to work extra hard? If I go to the gym and I work my muscles, what happens to my muscles? They get bigger, right? Now, that would be good for my biceps brachii, but that's not good for your heart. So a heart that over 20 or 30 years has to work harder, it had to suck harder because the person wasn't very active and wasn't helping their blood flow by moving their muscles, what happens? The heart starts to enlarge. And an enlarged heart is less efficient and less able to pump blood well. So you don't want to have an enlarged heart, right? There are some people who have large hearts naturally, but as far as the enlarging of the heart, you don't want your heart to become larger than it is because it becomes less efficient. Now, this problem with your valves, here's a cartoon representing your valves. So you've got the valves going through a muscle group, just a simplistic idea, but as the muscles squeeze, it pushes the blood up, but then the blood can't go back down because the valves are preventing it. Now, these valves can go bad. People can go to a vein center. People who have venous insufficiency, whose valves are leaky or problematic, are people who oftentimes stand a long time of the day. So they're working at Walmart, they're a cashier, they're a teller, they're at the bank, whatever. They're not moving their muscles very much in their legs. They're sitting there. The skeletal muscle pump is not doing much to assist in the movement of blood. Those valves are being asked now to work even harder against gravity than they should. And over time, those valves start to leak. And now blood starts moving backward, pooling in the leg. Now, even a person working at Walmart who stands there all day is still using his or her arms. So it's not usually as much of a problem for the arms as it is noticeable in the legs. Now, as those valves fail and blood starts pooling back in the veins, veins do what? They don't have much of a muscle wall. So when the veins overfill, the veins spread. So now you've got varicose veins or spidery kind of veins, and that's, an overall story of venous insufficiency. Maybe the valves are just failing genetically or because there's something wrong with them, or perhaps your lifestyle is not very active or you work in a job where you're standing a lot. So people wear support hose, right? And that gives more pressure on the muscles and essentially helps with this blood flow back to the heart. This is just a quick pictorial review of everything I've said. It's just a great thing to stare at for a moment. But you see on the right-hand side the three types of arteries: big elastic, medium-sized muscular, smaller arterioles, capillaries, small venules, medium and large-sized veins. Notice the valve in the medium and large-sized veins. Okay, just a couple more concepts to share with you today, and we won't be finishing this whole presentation. The last 20 slides or so basically are name that artery, name that vein. We've done that in lab, and you don't need to know any arteries or veins beyond what was on your list in lab. But I do want to talk about blood pressure, and I do want to talk about baroreceptors, and then I'll jump over to the very last slide that says what happens as you age, what happens to your vessels as you age. So blood pressure. Who in here gives or takes blood pressure? Anybody as part of their job? The old-fashioned way or with a machine? Both? Old-fashioned way? Okay. So what do you do? I mean, what's going on here? As your heart pumps, there is pressure being put on the inside of your vessels, and that's the idea of blood pressure, the pressure that's caused by your heart on the inside of your vessels. This rhythmic pressure is measured in millimeters of mercury by a sphygmomanometer. Sphygmom, remember, means pulse. So the portion of the blood pressure cuff that measures the pulse is the instrument called the sphygmomanometer. You're measuring two numbers, systolic and diastolic pressure. Systolic means contraction. Diastole, the stand apart or the relaxation time when the heart is not contracting. The two numbers, systolic over diastolic, anything over 140 systolic, your doctor is going to say that you have hypertension of some sort, high blood pressure. If you have a diastolic over 90, also you're going to be described as being hypertensive or having high blood pressure. Which of those two numbers would you be more concerned about having high? What if you went to your doctor and she said you have 130 over 95, only the diastolic's high, or you went and they said, you know what, you have a pressure of 135 over 100. Oh, is that backwards? Or you go in and it's 150 over 80. Okay, so the systolics alone is higher. Which one would you be more concerned about? Which one's your doctor more concerned about? A higher systolic or a higher diastolic? Diastolic, that's correct. Why? Right, this is the pressure on your vessels even when your heart is at rest. When the bear walks in the room, your pressure can go up to 200 over something, no problem. Your vessels can handle high pressure for a short while. But you would want them at rest to be as rested as possible. So you wouldn't want to have substantial pressure on your vessels in your diastolic periods of rest. Okay, so a higher diastolic suggests that your vessels are not as healthy, and a higher systolic is a little bit more tolerable. 120 over 80 sort of being an average of all individuals. In a younger group, it'd probably be lower, and it does get higher as we age, your blood pressure. Now, as I've said, the pressure is very high when the blood leaves out of the aorta, is quite regulated as it goes through capillaries, and I've told you is extremely low when it goes through veins. It almost comes back as 0. And finally, if you squeeze on the hose, vasoconstriction will always raise your blood pressure. So in this diagram, let's look at the bottom of this. I know it's small. On the bottom, it basically says aorta, arteries, arterioles, capillaries are in purple, small venules, veins, and back to the vena cava. As the blood is leaving the heart at the aorta, about 120 over 80, pulsing, systolic, diastolic, back and forth, back and forth, back and forth. When we take blood pressure, we normally take it from the brachial artery. It's not, directly at the heart, but it's pretty close by. So we look at the brachial artery. So the arteries, when you look at the arteries, the top and the bottom numbers, it's not the same, but it's not much different from what is represented by the pressure coming out of the heart. So it's a good way for us to measure how the vessels are doing. Now, as you move down toward the capillaries, right here, what do you notice? The blood pressure is much lower going into the capillaries, around 40 millimeters of mercury, but you also don't see a difference. There's very little pulsing, right? So that means that blood is going into the capillaries kind of as a steady stream. It's kind of moving into the capillaries under reasonable pressure and isn't pulsing strongly like it is out of the heart. The blood then goes through the capillaries, and as it comes out of the capillaries, it continues to drop. And eventually, as it comes back to the heart, is essentially 0. So that's the pressure that the vessels are experiencing as the blood travels through your body. Now, we could take blood pressure from the femoral artery, right? You can wrap a cuff around the leg if you needed to, and that's okay. You could do it around your neck, take the pressure of your carotid, but that's not a good idea because you have to kind of stop the blood flow for a while, and that's not good. So we wouldn't want to wrap it around your neck. We could, wouldn't be healthy. And we could take your pressure, I guess, if you could get in there and grab your aorta, right? Again, not a good idea. So we do it on the brachial artery. We can take blood flow being restricted to our arm for a short while. Now, I just want to make sure that we understand what blood pressure is. I've got like a couple slides. I'm going to be a couple minutes over, but I think we'll be okay. So when you take blood pressure, what do? You take the cuff, put it on the brachial artery, and you pump, 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 pump up the sphygmomanometer and the blood pressure cuff. And the idea is that you want to completely block the flow of blood through the brachial artery. So in this cartoon, what we're showing over here on the right, the vessel, the brachial artery, is completely blocked off. It's closed. So you pump it up. Your fingers start getting a little numb, right? The pressure is pretty significant on the cuff. And then start slowly releasing that pressure. Now, at some point, as the cuff gets looser, blood will start moving through your brachial artery. At first, only under systole, under the contractile phase of the heart. And as long as the blood is restricted in its flow, there was a guy named Korofkov who back in the late 1800s noticed that he could hear the blood moving through the vessel when it was restricted. So these are named in his honor, Korofkov sounds. You don't hear those Korofkov sounds at the beginning because the vessel is completely blocked. There's no blood traveling through it. And once the pressure reduces and the flow is restored back to normal, you also don't hear that pressure, or that Korofkov sound. So as you start to loosen the pressure, the first lub, you hear the sound at the stethoscope, right? You're listening for the flow. The blood's trying to come down through the brachial artery, and it can't, right? It's blocked. And then it starts coming through, and you're going to pick up that sound on the stethoscope. That sound you're hearing is the Korofkov sound, and it tells you that the blood is now flowing, but restricted. And as long as there's some restriction in the flow, you'll continue to hear the Korofkov sounds. The first Korofkov sound you hear is recorded as a systolic pressure. You'll continue to hear those sounds. The last Korofkov sound that you hear, or as the sound disappears, that represents the diastolic pressure. Okay, and that's really what's going on. You are not hearing lub-dub, lub-dub, lub-dub. Okay, in the stethoscope, you are not hearing the heart valves. You are hearing these unique sounds of restriction of blood flow through the vessel called Korofkov sounds. Now, one concept to share with you, and then a quick mention about what happens as aging. If you were going to design the cardiovascular system, and you had to put into place some warning signals or bells or whistles that would alarm the body that the pressure was too high or too low to sustain life, where would you put those warning signals, those sensors? Well, the good Lord put some baroreceptors in your aorta, kind of telling you about the pressure of your heart, right? Blood coming out of your heart. And also in the internal carotids. Think about those. Those would be the vessels bringing blood up to your brain. Kind of important that your brain's getting enough pressure. When those baroreceptors, receptors that sense what? Baro, pressure. If the pressure is low, right? If there's low blood pressure, and those baroreceptors are not being stretched, then there will be action potential. This is a fancy word for saying nerve impulses. Nerve signals will be sent to slow down your parasympathetic, but increase your sympathetic. Well, that makes sense. When the bear walks in the room, your heart rate goes up. So if your blood pressure starts to drop, your body kicks up your sympathetic signals, which tell your heart to beat faster, increase your blood pressure. Flip it around. If your blood pressure starts to increase and those baroreceptors are being stretched, now you're going to stroke out, right? There's too much pressure. Okay, now they're going to send nerve signals that are going to tell your parasympathetic signals to increase and your sympathetics to decrease. This is all part of your homeostasis. This is completely out of your control, involuntary, but part of how your brain and your heart and your blood pressure are being regulated. Now, not only is the heart measuring your pressure, but there are also chemoreceptors. They are measuring your chemical makeup of your blood. They're in the same place. So also in your aortic arch and up in the carotids, there are chemoreceptors. They're basically able to measure your oxygen, CO2, and acid levels. And they are also going to help dictate and speak to your heart, speak to your brain, and regulate your blood pressure. The next picture just shows you where they are. So these receptors, this is specifically for the baroreceptors. They are located in your aortic arch and in the internal carotid. Makes sense, doesn't it? Making sure there's adequate pressure to your brain and adequate pressure to the rest of your body out the aorta. You shouldn't also be surprised that the vagus nerve is involved in this. So the vagus nerve, number 10, is going to be receiving. Remember, the vagus nerve wandered all the way down into your heart and your gut and your organs and sent signals back about how your heart was doing. Well, what it's picking up are these baroreceptor signals, and it's reporting back to your brain how your heart is doing overall. While I'm here, name this vessel right here. Coming off the aortic arch. This would be the brachiocephalic trunk. Remember that it splits. This would be the right subclavian, the right common carotid, splitting into the external carotid, and there it is, the internal carotid. This would be the left common carotid, goes straight up the neck. And what's missing, the third branch off the aortic arch, would be the left subclavian. Just wanted to review that with you. The rest of this is just good old flow of arteries and veins, not for you to memorize, not for you to worry about. You already know your arteries and veins, just know the same ones. If someone is having angina, right, partial occlusion of a vessel, this is what they'll do. They'll go in and they will do a graft. They will reroute around it. So here's your coronary artery coming down. There's a blockage right here. That means that this part of the heart's not getting enough oxygen. So fine, we'll make another route. We'll take another route. We'll take a vein out of your leg and bring it around and bring now blood, bypass that clot, right? So we're going to bypass it and bring now total blood flow here. Or we can go with a little balloon and bring in a little machine, kind of a little balloon, and open up that balloon and now open up that atherosclerotic plaque. So we don't deal with angina anymore. We go in and we treat it. We open up those vessels, so we don't worry about that too much anymore. Finally, so if you hear a person having a double bypass or a triple bypass, they've found 3 arteries that are about to clog off, and they've rerouted blood around those issues. Very fortunate. If someone has a triple bypass, they're very fortunate because we found it before a major issue. Then the last thing I want to share with you is the very, very last slide here, and that is at the very, very end of this. Now, just a warning, there are arteries and veins in here you don't need to know, many, in fact. I overstuffed this presentation. So you only need to know the ones from the lab. Don't worry about the ones you don't need to know. And then finally, at the very, very end, what happens to your cardiovascular system as you get older? Boom, 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 boom. There it is. And as we can imagine, the vessels become less resilient. They tend to get a little bit crunchier, a little bit less elastic. And with that decreased elasticity, they don't stretch, pressure goes up, and more cracks can start happening in the arteries. And so over time, your blood pressure will increase with age, and your vessels become weaker, and the muscle wall can bulge and even rupture, leading to an aneurysm, and that would lead to a major bleed, which could be deadly. Okay? So that is the end of the content. I appreciate your patience going over about 5 minutes, but that's where I got with the morning group as well. If you have any questions at all, come chat with me really quick. But I think you'll find that there was a few extra added details here, but much of it I hope felt reasonably comfortable. Remember to do your 3 mastering assignments. The quiz that will help you is due by Wednesday tomorrow night, and test is on Thursday. Be very safe going home.